Okay, this video will finish up our tour of the early stages of vision, the basics that happen in the eye before more complicated processing happens in the brain. In the previous couple videos, we talked about some differences between rods and cones, like rods being better for dark vision and cones being better for color vision. Here we're going to explain one of the other main differences between rods and cones. So this was our original list of rod-cone distinctions, and now I want to explain why cones are so good with detail vision while rods are more honed for that big picture information or for detecting motion. To understand that difference, we'll actually have to look at the wiring within the retina, the way neurons are laid out as the signal goes from a photoreceptor to the exit of the eye and out towards the brain. So this diagram here, it zooms in on the retina, and this shows that there are some layers of other neurons, other cells in the retina aside from just the rods and the cones. So we've got the photoreceptors there in kind of pink and purple at the very back of the retina, the very back of the eye, farthest from the cornea and the lens. But as the light comes in from the front of our eye, it first passes through a bunch of other mostly transparent neurons before it finally hits those photoreceptors and actually, turns them on, starts neural firing. So once the light hits a photoreceptor, like a rod or a cone, that's when we get transduction. It goes pew, 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 sending a signal towards the brain. But notice that the photoreceptors here, the rods and the cones, don't send a wire directly out the optic nerve. They don't send a wire directly toward the brain. Rather, the photoreceptors are hooked up to these other nearby cells. Specifically, the photoreceptors are hooked up to these horizontal cells. We can kind of see this one in blue here, and these bipolar cells that you can see in a lighter blue. Then those bipolar cells are hooked up to amacrine cells. You can see in this very light blue a few of those hooking up kind of sideways there, and to these ganglion cells, the yellow big ones at the end there. And only at this point, only after the signal has passed through all of that, do the wires go out. The wires from the ganglion cell leave the eye. They send their axons out the optic nerve toward the brain. And notice in the picture here that really the fundamental pathway, the main pathway to get the signal out, goes from photoreceptor to bipolar cell to ganglion cell. Those other two, the horizontal cells and the amacrine cells, they're just sort of sideways connections to help out, but they're not part of that primary pathway out of the brain. So they're not going to be as important for us. It's maybe easy to remember that the bipolar cells are the ones in the middle. They're sort of middleman on the main pathway there because you can see they're sort of symmetrical with two poles, right? Almost like their arms reaching out, one to each side, two poles, two ends stretched off. So two sides or two poles would be bipolar. They're the middleman connecting those specialized photoreceptors that detect light to those much larger ganglion cells that actually send the signal off to the brain. This is just a flow chart I made to make it a little more clear without the jumbled up picture. So the main thing to note is that once the light comes in, remember it passes through all that junk on the way, but once it comes in and actually hits the photoreceptors at the very back, it's then going to go to bipolar cells, then to ganglion cells to get out of the eye. That's the main thing to remember. The main pathway of information, once the photoreceptors go off, just has a couple steps and then it's sent out to the brain. Those horizontal cells that we saw before, they're just a kind of sideways or lateral connection. They really help out by connecting photoreceptors. We'll see in another video why that's important and helpful, but for now, just think of them as support cells along the main pathway. Likewise, the amacrine cells, the other ones we saw, are just more lateral sideways connections, and they again help out by connecting bipolar cells and by connecting up ganglion cells. But the main pathway is still from a rod or a cone to a bipolar cell to a ganglion cell and out the optic nerve to the brain. Now, looking closely at this diagram, this is just another version of that same wiring, that same setup. You got the photoreceptors at the very top. That's the very back of the eye here. So light is coming in from the bottom in this image, passing through all the gunk, hitting the photoreceptors here, and then they send their signal out to these reddish bipolar cells, which then go out to these yellowish ganglion cells. Okay, they're not actually that color. This is just in the diagram. They've made them red and yellow, so we can tell them apart. So One thing to notice about the wiring here that I've kind of zoomed in on or homed in on with this little square here is that sometimes we've got multiple photoreceptors, like multiple rods, sending their signal through to a single ganglion cell. Like here on the far right, we've got 3 rods at the top, all of which hook up to that one bipolar cell, which then hooks up to that one single ganglion cell that sends its wire to the brain. That means if the brain gets an action potential from that ganglion cell, from that single wire, it doesn't actually know which of those 3 rods went off or in which combination they went off. This type of wiring is called convergence. Convergence just means when multiple earlier neurons all synapse onto one later neuron, like connect onto or feed into one later neuron. So just for example, we can see here a bipolar cell, a single bipolar cell can get info from multiple photoreceptors. That would be convergence. But also looking at this ganglion cell down here, the fact that it's like originating signals comes from 3 different photoreceptors means this ganglion cell also has convergent wiring. And since bipolar cells are really just kind of passing the info along, that means fundamentally when we look at those ganglion cells, some of them can trace all the way back to a lot of photoreceptors getting lots of convergence, while others might trace their signal back from only one or very few photoreceptors having little or no convergence. So we might even find a ganglion cell that hooks up to like a single cone and nothing else. That would be no convergence. Or if we have one that hooks up to a bunch of cones or a bunch of rods, that would be a lot of convergence. So just to make it clear, here's another diagram showing this same idea, the same thing. So again, we've got a bunch of rods and cones up here, a bunch of photoreceptors up here. So they've written this as receptors, R. Then we got the horizontal cells. Don't worry about those sideways connections, but we have a bunch of bipolar cells in green. And down at the bottom, these ganglion cells, which again send their signal out to the brain. Well, if we look at the 3 that I've highlighted in yellow here, the 3 rods that I've highlighted in yellow up top, if you follow them, you'll notice their input, if you follow the row, all eventually goes to the same ganglion cell in the lower right there. So the firing of that ganglion cell will be affected by the firing of any or all of those 3 rods that are connected to it. So it's not specific to input from just one rod. It has convergence. Now, each of our eyes has about 126 million photoreceptors in each eye, but only 1 million or so ganglion cells. So a lot of rods and cones relative to those larger ganglion cells. That means on average, a ganglion cell gets input through bipolar cells from about 126 photoreceptors. That means a lot of convergence. But remember, we have way more rods than cones. We have about 120 million rods. So in that case, a ganglion cell might get signals from, say, 120 different rods, whereas we only have 6 million cones. So a ganglion might get signals from 6 or fewer different cones. In fact, if we go to the fovea itself, in the fovea itself, where it's only cones, every cone has its own personal ganglion cell. So for cones, it's pretty common to get their own private wire to the brain. No convergence involved for a lot of those cones. Whereas convergence is very common with rods, and we can see there's a lot of convergence. A single ganglion cell might hook up to a ton of rods, like 100 or more. So rods tend to have way more convergence. And what this means in practice is that they get the benefit of more sensitivity. That means like a lower threshold, easier to pick up very, very, very faint light. This convergent wiring makes it easier to detect that tiny amount of light. And to understand why, let's play with this simplified diagram I've got here. What we've got is 5 individual rods. And for now, maybe we'll just ignore the bipolar neurons, the middlemen there, and just draw, eventually their signals end up at this one ganglion cell here in red. So we can see from this wiring, we've got some convergence. The one red ganglion cell gets input from 5 different rods, each rod in a slightly different place at the back of the eye. So each rod is getting light from a slightly different angle, meaning a slightly different part of the visual field out there. Now imagine, just to make up some totally arbitrary amounts, imagine the only way for that red ganglion cell to fire, it has a threshold, it'll only fire a signal to the brain if it has, let's say, 6 total input from its 5 hookups. That means that even a tiny faint light out there in the world might turn on, might hit each of those rods, let's say just a little bit, maybe just 2 units hitting each of them. Again, arbitrary units, but let's say 2 light hitting each of these rods. That means even though the light out there in the world isn't very bright, it isn't 6 strength by itself, this convergent wiring can kind of add up the input from those multiple detectors, which means that the ganglion cell will fire. It will have a response and send a signal to the brain, even from this very faint signal, this very faint light. So that wiring has given us really good sensitivity. We can detect very faint light, like just 2 units. Again, I'm making up arbitrary units here, even though that red ganglion cell needs 6 total to fire. Now, imagine instead, compare this to a different type of wiring where we've got less or no convergence. So here on the right, we've got 5 photoreceptors. This time, let's make it 5 cones, okay? But each one goes to its own private, like a single ganglion cell that we've drawn here in red. Again, we're skipping the bipolar cell that'd be in the middle, passing the signal along. But just like before, we could say a ganglion cell only fires if it gets 6 total input. Same rule as before to decide whether a ganglion cell fires a pew, 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 pew signal off to the brain. And in this case, if a really dim light comes on, a really mild light, like only 2 strength or something like that, then when that dim light hits each of those cones and turns them on just a little bit, just 2 strength, that means the ganglion cell is not going to get enough to fire, right? Each of those is not going to get enough on its own to fire. It doesn't reach 6, so there's no signal to the brain, no response in any of those ganglion cells, even though there is some light out there. The light is just so dim that this non-convergent wiring doesn't let us detect it. So we don't see it. We don't perceive light. We don't see anything. So by comparing these 2 wirings, convergent and non-convergent wiring, we can see that convergence has the benefit of increased sensitivity. It can detect fainter light. Cones, with their private non-convergent wiring, they kind of suck at sensitivity. But that's okay. There's a trade-off here. There's something those cones get in return. So cones, since they have less convergence, it actually gives them better acuity, meaning more precision, more ability to see detail and sharpness. That's why whatever you focus on, whatever hits the fovea, whatever you're looking directly at, is much clearer than the things off in your periphery. No one reads a book by focusing at the margin and reading the letters and the words off in your periphery, because you basically can't do it. So acuity, that sharpness, that's what's measured in like those standard eye charts that you see at an optometrist's office. But the real question is, why would a lack of convergence, why would a private wiring from each cone to its own ganglion cell give us better acuity, better detail vision? To see that, we'll try another diagram here, just to kind of play it out with a simplified diagram to see how the wiring might affect it. So at the top half of this picture, we can see a couple examples here. What we've got are some rods, 5 rods hooked up with their convergent wiring. Lots of rods to a single ganglion cell. Now, when that red ganglion cell fires, right? So if it fires a signal, pew, 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 off to the brain, that the brain gets this signal saying, oh, the ganglion cell went off. That ganglion cell went off. But notice that ganglion cell's signal might go off, might send that signal to the brain. It'll be the exact same whether the light came in, let's say in the upper left pattern here where it hits rods 4 and 5, or if the light came in a totally different pattern and hit maybe rods 3 and 5, or even a different pattern where it hits 1 and 2, or 1 and 4, or 1 and 5, or 1, 2, 3, 4, and 5, or any other combination. There's a ton of different ways that this one ganglion cell can go off. In other words, a ton of different circumstances, a different spatial orientation of light, all of which will look exactly the same to the brain because it causes the ganglion cell to fire in the same way. So with this wiring, the brain simply can't tell the difference. When the brain has to figure out what's out there in the world and precisely where things are that it's seeing, it's reconstructing that visual field, our whole visual experience, from just the pew, 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 pew of ganglion cells. And here it doesn't have enough precision to be sure where exactly the light was coming from. Now, compare that to the way cones tend to be wired, where we've got little or no convergence with their own private little ganglion cell, their own private wire going to the brain. In the rightmost ganglion cell here, if the rightmost ganglion cell goes off of these 5, we know exactly where the light was coming from, where in space there was something that we're seeing. And if the right 2 ganglion cells both go off, we know exactly how much light was where, and the brain can easily tell that spatial pattern of light from the other pattern, like when the 3rd and the 5th cone go off, and thus what the brain hears is the 3rd and the 5th ganglion cell going off, or any other pattern there. All of them are super specific. So if a ganglion cell sends a pew 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 to the brain in this case knows exactly where some light was out in the world. So this cone wiring gives us really precise and detailed vision. Less convergence means more acuity, better acuity, but with the trade-off of less sensitivity to very dim light. By the way, if you want a really fun application of this fact, you can experience it directly when you're stargazing. Like if you ever go camping where there's not a lot of light pollution and you're looking up at all those stars. There are some stars that are pretty damn dim, that are just barely detectable with the naked eye. A common example comes from the binary star system of Sirius A and Sirius B, which you see in the bottom right here. It's 2 stars in a single solar system, but one of them is way more dim. So normally, if you look at that part of the sky with the naked eye, you can easily see Sirius A, the bright one, but you can't see Sirius B. It's just too dim for the cones in your fovea to pick up such a faint light. They don't have enough sensitivity. Oh, but sensitivity is just what your rods are good for. So what stargazers and amateur astronomers will do is called splitting Sirius. They'll actually focus not on the star itself, but a little to the side of it. So instead of looking directly at the bright area, what you do is just look off to the side a little bit, which means that the light from that star is now going to fall into your periphery. It'll fall onto your rods, which have that nice convergent wiring for extra sensitivity. Suddenly, if you're focusing off to the side a little bit, you're going to see both Sirius A and that dimmer Sirius B. This actually works the same for any star that is just barely detectable by human eyes. It has nothing to do with just binary star systems. This is just a common one. But anything that's really dim that you can just barely detect, you'll be able to detect it better or detect more things if you have them hit your periphery, hit your rods rather than your fovea. Another fun demo to really show the difference between the fovea's cone kind of vision versus the peripheral rod vision might make it clear why we never read things off in our periphery. So in a moment, I'm going to show you a bunch of letters all in a row along the middle of the screen here. So all in a row where the letter on the far right is going to be this X. Okay, I've actually drawn the X here. What you're going to need to do for this demo is just focus on the X the whole time. The X off on the right. Don't look anywhere else. Stay focused on the X. Are you ready? Stay focused. And now without moving your eyes, keep your eyes focused on the X. How many letters can you identify to the left of it? Don't look over there. Don't cheat. While still focusing on the X, how far off into the periphery can you identify letters before it becomes too confusing or blurry to be sure? So don't cheat. Don't look off to the side. How many did you get? Okay. You can look now. You can look and see how you did. Most people only get a few letters next to the X, like the D, K, Z, and maybe the P. And pretty soon after that, accuracy drops off quickly and people's confidence drops off quickly. We have trouble doing it. Let's do another trial of the same thing. This time I'll put the X on the far left, okay? So once again, stare at the X, don't move your eyes, don't cheat, don't look to the right. It's going to be hard to avoid, but just keep your eyes on the X. Are you ready? Here we go. How many letters can you identify in your periphery next to the X before you start to lose it? All right, again, it's common to get the ones right next to it. So like you can look now, the T, P, G, maybe some more. But then people quickly become unable to see things further out in the periphery with enough detail to identify the letter. Like we might detect that a letter is out there on the far right, but we can't tell what it is. We have sensitivity, but we have no detail off there in the periphery. Very poor detail because of that convergent wiring. Finally, remember that cones detect color, while rods are black and white. Rods aren't good for color detection. we can actually show that we have relatively few cones out in the periphery compared to the fovea by showing just how poor your color vision is off to the sides. It's way worse than you think, way worse than you realize, because as you move through the world, your brain is usually filling things in, inventing color off to the sides, but you're not actually seeing color there very well at all, and you'll be inaccurate if you're forced to guess. So the easiest way to demonstrate this is actually just grab a bunch of colored objects with the same shape, like some colored markers or Sharpies or whatever. And then what you want to do for this whole demo is keep your eyes focused forward on something. Like the lady in this picture is staring at that little push pin in front of her and not moving her eyes to the side. You could focus on like a clock at the far end of the room or something else off in the distance, whatever. But you just want to make sure, don't look off to the sides. Keep your eyes focused forward for the whole experiment, the whole demo. And then you'll need a friend or a partner to do the testing. It's fine to blink while they do the testing, just don't look off to the side. Now your partner or your friend, they're going to randomly pick one of the markers without telling you which color they chose, and they're going to hold it up about 1.5 feet away from you, but directly off to one side. Like actually coming maybe even from behind your vision, kind of like in this semicircle or circle around you. Maybe start the marker off a little behind your field of view, and then bringing it in at that same distance of about 1.5 feet or so, bringing it in more and more forward, okay? So it stays at the same distance from the eye, but gets closer and closer and closer to the fovea. Now, as soon as you can see that the marker is there, as it comes from behind and actually comes into the slight side of your field of view there, as soon as you can see that there is a marker there, maybe you tell your friend, you might say, I see it. But then have your friend stop and maybe just try and guess the color they're holding. Like say out loud whatever your guess is, but don't look, don't see if you're right, and your friend shouldn't give you any feedback at this point. Now the friend can just keep moving the marker really slowly, but just slowly move the marker further towards the fovea, getting it closer and closer and closer to being in the fovea, but still at that same distance. And then you might guess the color at a couple different points along the way until eventually, as it gets near the front, closer to the fovea, you might be super confident what it is. At that point, if you're really sure what it is, then you can look, see whether you got it right or whether your answer maybe changed as it got closer to the front and you became more confident. And then just repeat the same thing. Try it again with another random color, a bunch of different colors. It's fine to repeat the same color to make it even less predictable. But again, every time, every trial that you do this, always keep your eyes focused forward and try to identify the color when it's still way off in the periphery. You'll likely notice your accuracy is really poor out there in the periphery. Even though you can definitely see that the marker is present, or you can see that it's moving when it first comes into the visual field, you won't know the color for quite a long time after that, and your accuracy will be much lower than if it's close to the fovea, close to being right in front of you. In fact, a 2020 study by researchers Cohen and Rubenstein, they showed that we often don't notice even massive color differences in what we see if it's out there in the periphery. So they did an experiment, brought people into a lab so they could focus directly, and all these pictures, when they flash one of these pictures in front of people, people are focusing right in the middle at that red dot. So they're shown one of 3 different versions. For each picture, they're shown one of 3 versions. Either the original picture with the proper coloring or one of these altered pictures where the coloring is all messed up in the periphery. In all these cases, they had people focus on that dot at the center. And when they do so, people often don't notice that the image has no color in the periphery, or if the color has changed a lot out there. Our brain still sees it as a normal picture. They don't notice anything wrong. We perceive it as no different than the original unaltered image. In this case, in this experiment, like for example, in one condition, they flash the pictures pretty quickly, maybe 1/4 of a second, which is actually about how long our gaze normally fixates on objects as we look around. Our gaze kind of bounces from place to place, and we fixate for about 1/4 of a second before moving our eyes around. And anyway, what they had is to make the participants focus on the center, focus on that little dot at the middle. They were told the task was just to say yes or no if a human face was in the middle of the picture that was flashed up there for 1/4 of a second. And in that condition, 75% of people totally missed the color changes. They didn't notice if there was no color in the periphery or a really different color in the periphery. And that's because their fovea, their color detectors, their cones, were at the middle where the color was correct. It wasn't changed right here in the middle where the fovea would be. So the weird pictures didn't look weird to them. The messed up color wasn't noticed by most people. The researchers actually argue that our brains are basically just extrapolating color in the periphery all day long as we look around and bounce our gaze around the world for brief little stops at various parts of the visual field. Our brain's just filling in the blanks. They put it this way. Under this view, they say, although each independent sampling of the visual world, so as our gaze bounces around the world, each independent sampling of the visual world lacks much color in the periphery, so we don't actually see color out there in the periphery, those samples get stitched together in the brain to create an experience of color across the visual world via this color percept integration. So basically we think we have the perception of color, we experience it, we think we're seeing color out there in the edges, but actually we're not ever directly perceiving color in the periphery. All right, that'll finish up this first topic for vision. So far we've limited ourselves to the earliest steps in the process and learned a lot about rods and cones, but in the next topic we're going to go a little further into that process and towards higher level perception.